We're moving on to the spinal cord, chapter 14. So continuing with the central nervous system, the spinal cord. So we previously discussed the brain and the spinal cord now together, making the central nervous system. So again, just this inferior section. And the spinal cord is this major, you notice the word for sensory afferent, as well as afferent motor. So the spinal cord is the major sensory and motor afferent pathways between the brain and the inferior part of your body. So sending sensory information up to the brain and sending motor signals down away from the brain. So it is that major conduit. But also these latter 2 points here, this is describing how the spinal cord, independent of the brain, the spinal cord on its own without the brain's response, is allowing the spinal cord to control reflexive functions of our body. You know, a simple example, if you hurt your hand, you have that reflex to pull away. That's being completely controlled by the spinal cord. So starting with this view here from chapter 14, we see in an adult, the spinal cord, of course, starts at the inferior tip of the brain. And as an adult, the spinal cord, however, only reaches down to about our second lumbar vertebra, that second lumbar vertebral bone. And that inferior tip of the spinal cord is called the conus medullaris. So we'll discuss the growth and development, the tissue, and a bit of the function of the spinal cord. So to begin with the length and development of the spinal cord, as an adult, the spinal cord we see actually starts superior to any of our vertebral bones in the cervical region, superior to any of those vertebrae, as well as the inferior tip of the spinal cord, as we just mentioned, it stops right about by that second lumbar vertebra. So we'll point this out. Superior to the actual vertebrae is that very first initial section of the spinal cord. And then just slightly below it, there's that first cervical vertebra. We learn the name as the atlas. And again, the spinal cord starts superior to the atlas. However, as an adult, the spinal cord only spans down just about to that second lumbar vertebra. Really just there between the first and the second lumbar vertebra is right about at the inferior end of the spinal cord. So the point is the bones of the vertebrae grow much longer than the actual spinal cord does. So seeing that here in the text, again, the superior end of the spinal cord is actually right at the occipital bone, that big hole in the occipital bone, the foramen magnum. So again, the spinal cord starts superior to our actual cervical vertebrae. And as an adult, again, it only spans about to that area between the first and second lumbar vertebrae. And that inferior tip of the spinal cord is known as a conus medullaris. So if we make a little sketch, it just comes to a point, and that inferior tip of the spinal cord is called conus medullaris. So it's now taking a look at the growth of the spinal cord. It reaches its full length very early in life, about age 4. However, the bones of our vertebrae, so those cervical, thoracic, lumbar, sacral, all of those bones of the vertebrae, well, they have several more years of growth and development beyond age 4. So it's truly that early in life, the spinal cord and the vertebral column are the same length. The spinal cord stops growing at age 4, it does not get any longer. And then from age 4 onwards, the bones of the vertebrae are growing and lengthening around the spinal cord. And that leads to that difference in length between the two structures. Again, as we already mentioned, the inferior tip of the spinal cord is known as the conus medullaris. But now descending down from that conus medullaris is this ligament structure called a filum terminale. And this filum terminale, it descends from the conus medullaris, and it ultimately connects down to the sacrum. So all the way down to the sacral vertebrae. And then ultimately, inferior to that, there's this ligament that connects onto the tailbone. And the purpose of this connection now is we have this inferior attachment point from our conus medullaris, and now we have this attachment structure securing it and attaching it onto the sacrum, and this is giving stability from the inferior side. It literally works as, you could think, like an anchor on the inferior side to help stabilize the position of the spinal cord. So sticking with that idea of stabilizing and protecting the spinal cord, we have several layers of meninges, and these are the very same layers of meninges that we discussed around the brain. So the dura mater, arachnoid mater, pia mater, the same layers that surround the brain, they also pass down into the vertebral canal, and those same meninges, of course, pass into the vertebrae to protect and stabilize the spinal cord. And as a note, these meninges even travel inferior to the spinal cord. Spinal cord stops by the second lumbar vertebra. However, the meninges travel down into the area of the sacrum. And that's what the S2 denotes. So the meninges protect the entire central nervous system and even some areas inferior to the spinal cord. Surrounding the meninges, superficial to that, there's the adipose, the fat tissue of the epidural space. And fat anywhere in the body is always a great cushion. So that cushiony tissue of the epidural space, this is found between the bones of the vertebrae and the first layer of the meninges, the dura mater. And again, anywhere we have fat, in this case, between the bones of the vertebrae and the dura mater, fat's always a nice protective, cushiony tissue. There's the fluid of the cerebrospinal fluid. And again, this liquid is supporting and cushioning the brain, and the same for the spinal cord. So that's reducing any possible trauma. And again, spelling out the orders. And the good news is just like the brain, the same orders. So starting at the more superficial dura mater, moving deeper into the arachnoid mater, the deepest layer, the pia mater. And now a unique feature of the pia mater, this is only found and it's unique to the spinal cord, these denticulate ligaments. These are now on each side of the spinal cord, right and left sides, branching out laterally from the pia mater. And ultimately, these extend from the pia mater all the way out towards the dura mater. And extending out laterally on each side, this now stabilizes again the position of the spinal cord and prevents it from moving side to side laterally somehow within our vertebral canal. So if we make a little sketch, if we imagine this is the spinal cord, and paired on each side, there's multiple denticulate ligaments. And again, having attachment on each side would prevent the spinal cord from somehow moving laterally side to side inside of the vertebral canal. So just putting that together, again, the similar meningeal layers of the brain protect and surround the spinal cord as well. And in fact, these tissues of the spinal meninges on the superior side, they connect all the way up with the same meninges from the area of the brain. On the inferior side, we just pointed out that filum terminale, that ligament that connects down to the inferior side. So we have attachment from the top. We have that security attachment on the bottom. And all of those denticulate ligaments we just mentioned, all on the lateral sides, all to maintain the position and stability of the spinal cord. And just a representation here, we can see the bones, of course, give a level of protection. Deep to the bones, represented in yellow, there's the epidural space. The first layer of the meninges, the dura mater. The second layer of meninges, the arachnoid mater. And then the deepest third layer, the pia mater. And don't forget that any of these open spaces here, such as the subarachnoid space, would be filled with the liquid cerebral spinal fluid. So now penetrating all of those layers of protection to reach the actual spinal cord, and we'll start to take a look at a horizontal section, a horizontal slice through the spinal cord. And we'll discuss, of course, the location of the superficial portion of the spinal cord composed of white matter, and the deeper portion of the spinal cord composed of gray matter. And we have this nice mnemonic here, DAVE. This is now describing the function of the spinal cord. So the activity of different parts. So if we take a look at this DAVE, Dave, the function of the spinal cord, Dave applies to both the gray matter and the white matter sections. So we're describing the dorsal portion of the spinal cord is carrying afferent information. Or another way to say it, the synonyms, the posterior side of the spinal cord is carrying sensory information. So in this case, the dorsal sections of the gray matter or white matter would carry sensory information up the spinal cord to reach the brain, whereas the opposite side, DAVE, the ventral portion of the spinal cord is carrying efferent signal downwards. So from the brain, that efferent signal traveling down the ventral side of the spinal cord, or again, another way to say this, the synonyms, the anterior side of the spinal cord is carrying motor signal down the spinal cord. So that's the DAVE, kind of the very simple introduction. So now we'll go ahead and take a look at the details of the different sections of gray matter, as well as the different sections of white matter. So the gray matter, the deeper portion of the spinal cord, and again, just as a refresher, gray matter is largely a collection of the nerve cell bodies, the portion that contains the nucleus, the cell bodies, as well as complete interneurons. And this is a full neuron from dendrites to synaptic terminals that do not have myelination. So they appear still as that darker gray matter appearance. And in the gray matter, we have these three principal areas known as horns. So there's a dorsal gray horn, a ventral gray horn, and a lateral gray horn. And again, just applying that rule, DAVE. And let's see what this now applies to. The dorsal part of the spinal cord carries afferent information. So if we take a look at the dorsal horn, the dorsal gray matter of the spinal cord, it contains the cell bodies, because that's all this term. Nuclei are just simply cell bodies that are in the central nervous system. So the cell bodies of somatic and visceral sensory neurons are located and clustered together in that dorsal gray horn. And as we said earlier, The dorsal portion of the spinal cord has afferent function, and we're just spelling out specifically the cell bodies of somatic sensory neurons and visceral sensory neurons. Both sensory functions, both afferent functions, have their cell bodies clustered in that dorsal gray horn. So on the opposite side, the ventral gray horn is going to have a efferent or motor function. And here we are. So in the ventral gray horn, in the anterior gray horn, we do have, of course, the cell bodies of, in this case, only the somatic motor neurons. So cell bodies of somatic motor neurons. So that definitely fits with the ventral having the efferent function, and specifically its somatic motor function. Those cell bodies are clustered in the ventral gray horn. So there is now this third horn called the lateral gray horn, and you know it doesn't fit with our nice little Dave mnemonic. The lateral gray horns, now you notice, they have the cell bodies of the visceral motor neurons. So the visceral motor neurons, the involuntary motor function, those neurons have their cell bodies grouped in the lateral gray horn. And unfortunately, there's not a little nice mnemonic to wrap up all three sections. But definitely the dorsal with the sensory function, the ventral with the motor function, and we just want to be specific about somatic motor versus visceral motor. Now, there's a great figure in the textbook we'll skip to. This figure right here shows the very same thing visually. So we can see an anterior gray horn, the little bump of a lateral gray horn, and we have the much longer posterior gray horn. You could always interchange the words posterior and dorsal, anterior and ventral. You could always interchange those words. So if we look on the other side, the anterior gray horn, this is shown to contain only somatic motor cell bodies grouped in that anterior gray horn. And again, the ventral or anterior part has a efferent function. Specifically, it's somatic motor. The posterior gray horn, you see, has both somatic and visceral sensory cell bodies. So the dorsal part has that afferent function. So again, DAVE is very helpful. And then lastly, we see the lateral gray horns off to the side. The lateral gray horn, you notice, only has the visceral motor cell bodies clustered together. So let's continue with the gray matter. We have this midsection here known as a gray commissure. And as you can see, it's a connection between these two different halves of the gray matter. So we have that description here, the gray commissures. Again, a place where interneurons are crossing from the right versus left sides of the gray matter. And it has that communication between the two halves of the spinal cord. And a final note about the gray matter. Those lateral gray horns off to the side, they're not found. They are not found in all sections of the spinal cord. They're only limited to the thoracic segments of the spinal cord and the more superior lumbar segments of the spinal cord. Whereas the anterior gray horns, the posterior gray horns are all throughout. The lateral gray horns are limited to just these areas here. So moving on from the deeper gray matter, there's the superficial white matter. So this is at the surface of the spinal cord. And the simple term for the areas of white matter are the columns. And you see the description, just simply lateral, anterior, posterior. So those three areas of the white matter columns. A single column itself is made of multiple tracts. And again, that term, a tract, this is a bundle of axons, but by definition, tracts are only located in the central nervous system. So that bundle of axons, that's what comes together to make a tract. And again, Dave applies to both the gray matter and the white matter. So that would mean the function of a anterior white column, an anterior white column, the ventral or anterior white column would definitely have a motor function. And the posterior white column, the dorsal white column, would have a sensory function. So again, Dave is going to be very helpful. So just simply put, again, the anterior white columns just based on location. And of course, they're paired for each side, right and left portions. And then moving on from the spinal cord itself, taking a look at now this clinical application. So common treatments that involve the area right in the vicinity near the spinal cord. So this first one was known as a lumbar puncture or a spinal tap. The point of this procedure is to now insert a needle into the subarachnoid space, into the meninges directly surrounding the spinal cord. And you notice how this is done between the more inferior lumbar vertebrae. And this is done for safety. We said now a few times the spinal cord ends by the first and second lumbar vertebra. And inserting a needle in this area inferior to the spinal cord is done for safety, so not to risk injury to the spinal cord itself. And inserting a needle just into the area of the surrounding subarachnoid space, this is done to take out a sample of just a few milliliters of cerebral spinal fluid. And this would be done to test for any traces of infection that may be in that fluid. And if there is infection in the cerebrospinal fluid, that is a way to now identify infection that's in your central nervous system. If there's infection in the cerebrospinal fluid, that infection could possibly travel around the area of the brain and the spinal cord. And this is a way to, of course, verify that. Whereas now epidural, or also known as a spinal block, this is now an injection. So whereas a lumbar puncture is taking out a sample of fluid, an epidural is now an injection of anesthesia. And again, this is not going into the spinal cord directly, but in this case, into the surrounding adipose epidural space. And this infusion of anesthesia into this area, if done timely and correctly, this could really now numb the area, especially in the pelvic region. And this is commonly used to reduce the pain associated with delivery, with childbirth. So the epidural, again, would be done in this similar location, inferior to the conus medullaris, inferior to the tip of the spinal cord. The placement of the anesthesia into the epidural space, and that would diffuse into that posterior area of the spinal cord and numb the sensations of pain. So an example, you can see this illustration from the textbook, and we can see that third lumbar vertebra for placement. And this is showing an example of the position of a lumbar puncture needle. So as we just mentioned, going into the subarachnoid space, and it's done to take out a sample of cerebrospinal fluid. So that has to be inserted, of course, quite deep into the body to get that sample. Or, for example, in terms of treating a woman getting ready for delivery, you can see again that proper placement and insertion, of course, of that needle to infuse anesthesia in terms of the epidural. So moving towards the end of this section, we'll just go ahead and omit any of this from lecture. This is all going to be seen in lab, so don't worry about this section at all. In lab, we have studied the nerve structure. And at this point, we have now exited out of the spinal cord. And by definition, nerves are in the peripheral nervous system. And we're now just looking at the structure that holds a nerve together. So we said the definition a few times. A nerve is a bundle of axons located in the peripheral nervous system. And we're starting to see the layers of connective tissue that actually hold and bundle all of those axons together. So it'll be nice to start at the smallest scale. We can see here we have an axon in the peripheral nervous system. The myelination is afforded by a Schwann cell. And then now wrapped around the Schwann cell, wrapped around the myelination, this deep layer connective tissue is called the endo. Notice that prefix, the deepest internal layer, endoneurium. So wrapping around each myelin sheath. We have several of these now wrapped axons bundled together, and the unit's called a fascicle. So now this bigger bundle, the entire fascicle, one fascicle is now wrapped in perineurium. So that's the second layer of connective tissue bundling the fascicles. And now you notice multiple fascicles grouped together, and the entire surface of the nerve is now wrapped in the epineurium. So that outermost layer. And if you remember, we just covered the muscle section when it came to bundling and the structure of a skeletal muscle. It was the very same prefixes. Epi at the surface of the structure, the peri prefix bundling around fascicles, and at the deepest layer, the endo. So perineurium, bundling fascicles, multiple fascicles grouped together and wrapped in the epineurium. So all of this comes together to bundle a nerve structure. And of course, we see these nerves branching in and out of the spinal cord here. However, soon after nerves branch out of the spinal cord, they then interconnect. And that typical trait of bundling and interconnecting nerves is known as a plexus. And it's just noteworthy to point out that we have what's called the cervical plexus we've seen in lab, brachial plexus. However, in the thoracic region, you notice how there's just individual thoracic nerves that do not connect. So in this area, there is no plexus. There is no thoracic plexus. Whereas you see once again in the lumbar region and down here in the sacral region, we're back to having plexuses. But for the thoracic region, there is no plexus, does not exist. So from the beginning, we described that the spinal cord can control our reflexive functions. And again, this is an example of how their spinal cord could process and respond to stimulation independent of brain function, without brain function. So to spell out the steps involved in what's known as a spinal reflex arc, and our reflexes are these very fast, it could occur within 40 milliseconds, this very fast motor response to some incoming stimulation. And pain is always a good example. You hurt yourself and you quickly pull away. The 5 steps involved in a reflex arc are spelled out here. So step 1 is a sensory receptor, a cell that's detecting whatever that stimulation is. Pain, hot temperature, great example. The receptor in turn will signal to a sensory neuron, or we could say afferent neuron. And now we're going to carry that sensation towards the spinal cord. Sensory signals always go towards the CNS. In the gray matter of the spinal cord is where we see this integration step. This interneuron is going to process that incoming signal and then signal to a motor neuron. So now that efferent motor neuron will send that motor signal out away from the spinal cord, and it's now going to ultimately control what's called an effector. This is just the organ that's now going to be responsible for that reflex. So to see it on a diagram, this is a great example here. Step 1 with the receptor, and definitely pricking your skin, hurting yourself. That's a good example of this pain reflex, this withdrawal reflex. So the receptor signals to the sensory neuron, and we're now carrying, you notice sensory information going into the dorsal part of the spinal cord. The dorsal part receiving that afferent or sensory information. Step 3, the processing step occurring inside the spinal cord via the interneuron. The interneuron signaling, you notice, to a motor neuron at step 4. And now you notice here, the ventral part of the spinal cord is going to carry that motor signal away. So DA and then VE, the ventral part now having that motor function. And now we have the motor neuron signaling, in this case, to an effector. And step 5 is we have the reflexive response. So you injure yourself, and very quickly, using the spinal cord, we have that reflex to pull away. So great example of a reflex arc. Now, of course, signal does go to our brain, and we become aware of what happened, but the actual response and movement was completely controlled by the spinal cord working independent of the brain. We'll move forward now with this section going into the autonomic nervous system. And again, that's just another name for the visceral motor division. So meaning the involuntary level of control, the only way that we can control smooth muscle, cardiac muscle, and any of the glandular structures around the heart. So of course, we've already went through what it means to be a sensation, and sensory is divided into somatic and visceral sensations, the area in which we detect that stimulation, where that sensory input's coming from. And then we have the two types of motor functions. There's the voluntary conscious control that only applies to skeletal muscle, and that is somatic motor. And again, this involuntary autonomous function, the motor signal to help stimulate the pacemakers in the heart, for example, stimulating smooth muscle and blood vessels, your iris, and any of your glandular features, sweat glands, for example. That's all this visceral motor division. And again, we've already touched on the fact that there's generally these two major parts of the visceral motor division, sympathetic and parasympathetic. And overall, they have opposing activities. And as an example, one would slow down the heart rate in the case of parasympathetic, and the opposite, sympathetic, would speed up the heart rate. So they typically work against one another, but still it is that autonomous and voluntary control. So we'll start taking a look at the sympathetic division, and you see an alternate name, thoracolumbar. And this is in relationship to the location and the connection to the spinal cord. So the spinal cord, thoracic, and lumbar regions have an interaction with the specifically sympathetic portion of the autonomic nervous system. And again, taking a look at its overall effects, especially this bullet point here. So increasing our blood pressure, our heart rate, so our heart's beating faster, beating stronger, higher pressure to circulate blood. Also, you notice the ventilation, our breathing rate is increased. So truly, as we're using more energy, being more active, and the common phrase related to sympathetic is fight or flight. As we're using more energy and being more active, this will definitely stimulate increased activity of the sympathetic nervous system. And just a simple way I'd learned this years and years ago, the 4 F's, 4 activities, starting with the letter F. And when you do these, they definitely all upregulate your sympathetic nervous system. So you notice flight, as in running, fighting, right, being really physical, aggressive activity, feeding in terms of, if you had to actually hunt and chase down food, another physical activity, as well as the sexual climax. So all of these would definitely stimulate this increased activity of these various organs and systems of your body. It can lead to feelings of energy, euphoria. Okay, so this high level state of function, the sympathetic nervous system. But of course, we want to focus more on the anatomical structures. And we'll also see now the sympathetic nervous system has this interaction with the adrenal gland. And we'll see a illustration of this. But directly superior to the kidneys, there's what's called an adrenal gland, or a newer term, suprarenal gland, superior to the kidneys. And there's a direct neural interaction between the spinal cord, in this case, and the adrenal medulla. So whereas many other nerves related to the sympathetic nervous system, most of them have an interaction with what's called the sympathetic chain ganglia. But in the case of the connection between the spinal cord and the adrenal medulla, it bypasses that connection with the chain ganglia, and there's a direct nerve supply from the spinal cord going right into the deeper portion of that adrenal gland. In a general term, the deeper portion of many organs is called the medulla. So we could draw that in. It's that deeper area of the adrenal gland itself, the adrenal medulla. And our spinal cord, again, somewhere between those segments of T1 to L2 segments of the spinal cord, we have that direct nerve interaction leading right into the adrenal medulla, bypassing the chain ganglia. And ultimately, the effect is we see that our nervous system, specifically the sympathetic nervous system, will stimulate now the release of hormones from that adrenal medulla portion. And hormones entering the bloodstream, and specifically the name of the hormone is adrenaline, that'll of course increase the duration of that sympathetic signaling. The nervous system is very fast and rapid with a brief duration, but now directly controlling the endocrine organ, that of course will lengthen the duration of these effects. It'll give you an adrenaline surge. Whereas now, in contrast, the parasympathetic nervous system, this is another major part of the visceral motor portion, the autonomic nervous system. And we see an alternate name here, craniosacral. And the reason is now, in this case, a few cranial nerves of the brain are involved in sending parasympathetic signaling, as well as a few of the sacral nerves of the spinal cord. So we notice this is a very different location when compared to sympathetic. And we'll, of course, go through all of those anatomical differences. A quick look at the functions. So organ systems that are stimulated by the parasympathetic nervous system would include the digestive system, the urinary system, or your kidney function specifically. Whereas overall, this would now promote more of a relaxed, resting state for the body. In fact, a common phrase is rest and digest. So this would, of course, decrease our heart rate, for example, decrease our ventilation, and in a more relaxed state, our digestive system actually will work harder, as well as we have increased function of our kidneys. So again, overall, kind of the opposing activity of the sympathetic nervous system. And now getting into the alternate names and specifically the anatomical locations, the sympathetic or thoracolumbar division for the autonomic nervous system. Again, it has this interaction in its motor pathway. Remember, both of these are visceral motor pathways. And in the case of sympathetic, visceral motor pathways exit out of the central nervous system, somewhere between T1 to L2 segments of the spinal cord. Whereas the craniosacral division, parasympathetic, as we already pointed out, there's a few cranial nerves, 4 cranial nerves that carry this parasympathetic visceral motor signal, as well as there's 3 pairs of sacral nerves that also carry that parasympathetic visceral motor division. And you notice again, there's a very different location in terms of their anatomy. So this is a helpful illustration. As well as this chart here, it spells out a number of key differences. And we will come back to this because we'll first just organize this in a chart. So that's what we have here. Looking at the structural, the anatomical differences between these 2 visceral motor pathways, sympathetic and parasympathetic. So we'll just go through the chart and then we'll come back to some of these descriptions. All right, let's continue with the chart. So looking at this first row, what's meant by regional distribution? These are the locations where this visceral motor signal will exit out of the central nervous system. So of course, motor signal always travels from CNS towards the PNS. So we need an area where that motor signal is leaving the central nervous system. And we notice there's these different locations. The sympathetic visceral motor signal travels down the brain to the spinal cord, and it will exit the spinal cord. Again, there's that term thoracolumbar. And specifically what that means is within the spinal cord, between the thoracic and L2 segments of the spinal cord, so T1 to L2 is that location for sympathetic motor signal exiting the central nervous system. A very different location for parasympathetic, the name craniosacral, and we notice is specifically cranial nerves 3, 7, 9, and 10, oculomotor, facial, glossopharyngeal, vagus nerves, and sacral nerves, sacral nerve 2, 3, and 4, just commonly called S2, S3, S4. There's now what's known as these preganglionic and postganglionic neurons. And each of these nerve cells, if we make a little sketch, a nerve cell will have that long, slender axon portion. And we see that there's a sequence typically. There's a preganglionic motor neuron that has its axon, and then the second one in the sequence, a postganglionic motor neuron. And we're really looking at the length of the axons. So we notice for the sympathetic division, the preganglionic axon has a shorter length when compared to the parasympathetic's preganglionic axon. And now the postganglionic axon's length, we notice for sympathetic, it has a longer length when compared to the parasympathetic. So just to see some of this information visually, we'll start back at the top, the regional distribution. And that's the figure here. You notice we're looking at the sympathetic visceral motor pathway. And again, between the spinal cord segments, T1 to L2 segments of the spinal cord, that's where the sympathetic visceral motor signal exits out of the central nervous system. Looking at the length of the axons, and you notice the color coding down here on the chart. So the preganglionic axon seen in the red color, and you can see it has a relatively short length to it, whereas the postganglionic, or also just known as ganglionic neuron, here in black, it has a much longer length to its axon. And we could also notice that these two different motor neurons, the preganglionic and the postganglionic, they have this interaction at the sympathetic chain ganglia. So that's the typical case. The exception, however, is here for the adrenal medulla. So there we are, superior to the kidneys, the deeper part of the adrenal gland. And you notice here illustrated in red, one long single axon that spans all the way from the spinal cord to the adrenal medulla. So it's the one exception where there's not a pre and a postganglionic neuron. Whereas now in comparison, looking at this view for the parasympathetic division, we see first of all, you notice cranial nerves 3, 7, 9, and 10. And here it is just using the Roman numerals. And sacral nerves S2, S3, and 4 carry this information. So the regional distribution is passing through cranial nerves and sacral nerves, a different location from sympathetic. If we look at the length of the motor neurons, this time the preganglionic neuron has a much longer length when compared to the sympathetic counterpart, whereas, of course, in black, the postganglionic neuron has a much shorter length when compared to the counterpart in the sympathetic nervous system. And we notice here, there's no interaction at all with the sympathetic chain ganglia. So there's no interaction ever here. So we're able to see that visually, as well as the diagram we'll go back to. So let's continue with a few more traits here. Well, we just said this one here, the connection with the chain ganglia, usually for the sympathetic division, never the case for parasympathetic. And then the final difference we'll focus on is looking at the postganglionic neuron. You realize the type of neurotransmitter produced. And for the sympathetic division, we'll just use this abbreviation here, NE for norepinephrine. And in parasympathetic, there's ACH. We'll use that abbreviation for acetylcholine. So we'll finish up with this chart here. This shows a lot of that same information visually. So you notice the length of the axon, the preganglionic axon. Fiber just means one axon. Sympathetic, it's shorter. Parasympathetic, we can see it's longer. That's the reverse when you look at the postganglionic axon length. Sympathetic has a longer one. Parasympathetic has a shorter length. We can see sympathetic has that direct connection into the adrenal medulla. It has connections typically with the sympathetic chain ganglia. So the point is we usually have the interaction with the chain ganglia. The exception is leading directly into the adrenal medulla. Parasympathetic never has any interaction with the sympathetic chain ganglia specifically. And then lastly, looking at the postganglionic neuron, the specific neurotransmitter produced, we can see here for the parasympathetic, the blue coloring is representing the release of acetylcholine. Whereas here for the sympathetic division, the postganglionic neuron is releasing norepinephrine at its synaptic terminals.